So I will talk a little bit about our research on autism, or autisms with a big S, because it's a very heterogeneous condition that you will see. So as François said, I'm a basic researcher. I'm interested in something that is strange, that is the social brain, how we can communicate each other, how we can understand our feelings. And so people say, why are you working on that, and why you, especially as a geneticist, you know. And as Pasteur used to say, there is no applied research, there is application of research. So I'm a basic researcher, but I'm also interested in public health at Pasteur. So we are interested in the disorders that could affect the social brain. And one of the main disorders that can affect the social brain is the Autism Spectrum Disorders, but also other disorders like ADHD or epilepsy or dyslexia can really affect the way you can communicate with each other. So to do that, we are many in the lab, and many people, really beautiful people, that are geneticists, psychologists, psychiatrists, neurobiologists, people listening to mice, and they are all in the same lab, sharing the same cafe machine, and we can talk about the social brain together. So autism spectrum disorder is 2 diagnostic criteria. It was 3 before, and now it's 2. The lack of social interaction and the presence of stereotypy restrictive pattern of interest. Before, the language was one of the key points in the diagnostic, but you have some people who have almost no language, like in a phenotype, but people who are language, and that's what we call Asperger's syndrome. So the prevalence is about 1%. So I know in US, people think there is an epidemic of autism. It's probably more the public awareness and the medical awareness and the fact that we are using more broad diagnostic criteria. But still, there are people with autism, and more than we thought 10 years ago. There is more males than females in autism, about 4 males for 1 female in the phenotype, the more severe form, and about 8 males for 1 female for the Asperger. But I think we are very bad in diagnosing Asperger in girls. And we have some evidence that there are maybe like a camouflage of the girls with Asperger. They are a little bit more shy, anxious, and they don't have the full spectrum of Asperger syndrome. It depends where you are in the world, but the intellectual disability is something that is really a comorbidity that is often present in autism. Before 2000, about 2/3 of the patients have intellectual disability, and now after 2000, about 2/3 have no intellectual disability. So you can see that there is a difference in the spectrum of autism before and after 2000. There is almost no therapy, and there are some teaching strategy, and so we have to do something for the people. So I love the double helix, but I'm this kind of geneticist who think that there is not only genes and only DNA. My dream is to work on epigenetics, and when I get older, I will work on environment probably. But I still have my homework to do on genetics. So I think that's one of the information that could tell us about autism. So the phenotype is what you see, okay? And I don't know who is aware about the DSM-4 or the DSM. Yeah. So the DSM is the category of 1 page for ADHD, 1 page for schizophrenia, and I think genes don't care about the DSM-4. They probably never read it, and but every researcher tried to find 1 gene for 1 page of this book. So I think it's not like this. We are really looking at patients, like haute couture way of doing it at Pasteur. So we are looking, of course, at patients, but we're also looking at the unaffected siblings, the parents, the uncles, the cousin, and trying to see what really segregates autism, but maybe other more dimensional aspects. And we are looking at the people, not at a book or a page in a book or a disease. I was trained as a biochemist. I still think that biochemistry can really tell us a lot about biomarkers for autism. We are doing a lot of brain imaging, and we'll have no time to talk about it. And we are starting to do to derive neurons from patient cells trying to get at least access to some of the defects that are in the brain of people with autism without going into their brain. So 3 steps to identify genes for autism was the first step. To understand the mechanism of autism is the second step, which is more difficult, and to find treatments. So to identify genes for autism, so this is it. This is my life, and now I'm looking at the student who is looking at the DNA, but still I'm still looking for DNA. And these are the difference between you and me. I mean, it's about 1 letter, so we have 3 billion letters, ATGC, very simple, ATTTGCTT, and 1 on 1,000 is different between you and me. Some are what I call green, they don't make any difference. Some are yellow, it makes that you're blonde or brunette, which makes not a big deal. And some variations are really high risk for disease. Okay. The problem when you sequence the DNA, you don't have the color. So you need researcher, you need a really interdisciplinary team to see if it's green, yellow, or red. And we call this variant SNPs, so it's a single nucleotide polymorphism. There are other types of variants, like copy number variants. So sometimes you have a loss. For this kid, for example, he lost 5 million of letters. Okay, and you can have gain of DNA also. And again, it could be green, it could be yellow, it could be red, it could be neutral, not so much risk, and a higher risk for autism. So this is your brain at birth, and you have almost the same number of neurons when you were at birth and now. But during your life, it was, especially during the first 3 years of life, the connection were amazingly increasing, and these connections are called synapses. And at the synapse, this is a very simple scheme of the synapse. It's more complex normally. And you have this postsynaptic with receptors and the vesicles. And our work is to look at this synapse in autism. So 10 years ago, we found these first genes in autism that were affecting these cell adhesion molecules at the synapse called neuroligin. And then we found SHANK and we found neurexin. That was the first link of a gene with autism, and this was the first link of a synapse. That synapse was related to autism. So just to give you an example, this is the first paper we published with 1 boy here. So square of boy and circular girls. This is a boy with high functioning autism. A guy with autism with a normal IQ. And his brother, he has Asperger's syndrome. He wanted to be a nurse, then he wanted to be a taxi driver. He knows all the streets of Göteborg, so it was where he lived. And they all, both, they have, instead of having TTT, they have TTTT, 1 extra T. And this extra T is creating a stop codon. So normally the protein is like this, and it's a short protein. Just 1 base, and you have autism or Asperger's. So that was the first discovery. We sequenced about 150 patients at that time. We found only 1 family, and we looked at this neuroligin 3, and we found another family with this mutation arginine 451 cysteine. So we were there. It was really not the gene for autism. It was 1 information, but it was an interesting information that this molecule here, neuroligin, were involved in autism. So why not this one? Why not this one? Why not? So like an Aryan thread, if you have this information, can you find the other gene? So that's what we found. That's what we've done. And this is neuroligins, and we found that SHANK is involved in autism. So of course it's 1 slide, but you can imagine it's a lot of work from students and postdocs that are working on it. So we found SHANK, and then we found these are the neuroligins, and we found neurexins are involved in autism. So this scheme that maybe was a little bit complex is now a little bit more simple. You have neuroligin, SHANK, and neurexin. And they are making like a social synaptic network in your brain. So this is more simple. It's exactly the same. I mean, if you want to go from Paris to New York, you can be stopped there, you can be stopped there, you can be stopped there. But at the end, you will not go to New York. Okay, and so that to find this map, to find this pathway was the first way to look at autism in a new way and trying to get targets to find what's going on with autism. So the Iron Freight, 10 years after, it's this one. So this is the last review we published with chromosome 1, 2, 3, 4, 5, and so on. And these are all the genes that were found mutated in autism. But most of the time it's one gene, one child. Okay, so that's it. The news is that there are many genes for autism. The good news is that they converge in some pathway, like chromatin remodeling, mRNA translation, and synapse. And here you have all the proteins that are binding partners, and they all play a role in the synaptic function. So we are still trying to find new genes, and I have a colleague here in New York, in NYU, Gordon Fischel, who had a mouse who was grooming a lot. And he said, oh, this is an interesting gene. Are you working on this gene? And I said, yeah, we found a child with autism, and he has this a deletion. He has a loss of one copy of this gene. So, and that was one mechanism that could lead to autism. And now we found new genes that are not published, where these genes are expressed in the auditory pathway. And this, when we went back to the patients, they had this hyperacusis that some people with autism have. So, to understand the mechanism, I told you that the neuroligands are the synapse. What is amazing that if you overexpress a neuroligand in a neuron, it will create synapses. And you don't have to be a neurobiologist to see that if you introduce the mutation we found in this Swedish family, you don't have any more synapses. Okay? So it's really the functional. We have some idea now on the function of these proteins. It's again a lot of work with Schenck 3, Schenck 2, and the message that there is a problem with the synapse. It could be more synapses, could be sometimes less. It's the regulation of the synaptogenesis. So to go further, we wanted to understand what's going on really in the brain, not in the dish with a neuron. So we made a mouse with Tobias and Michael that is deleted for Schenck 2. So just like in the human condition. And that's Elodie. I mean, she's at Pasteur Institute, and she was listening to baboons in Nigeria 6 years ago, and now she's listening to mice at the Pasteur Institute. And what she found, that's the first thing she found. This is a wild-type mouse. If you have never seen a wild-type mouse, this is the usual behavior. So we looked at the social interaction, and this is a male, and we will introduce a female in estrus in the cage. So this is a wild-type male, so normal male. So if you have never seen a male and a female in estrus in a cage, this is what's happened. At least at the Pasteur Institute. I think it's quite universal. Okay, he's a man and she's a female. Okay, that's the normal behavior. Okay, this is a Schenck 2 male. So he has no problem of olfaction. He has no problem of memory. He can learn. I mean, but it looks like the social brain is different, and he has more interest. So you see, it's not that he's not interested, but it looks like it's really different. And so we made a lot of statistics. I mean, it's not only on this video that we. So you see, he will follow the mouse, but it's very. I mean, his behavior is totally different. And when I found this gene 10 years ago, I didn't think it would be more like you see, it's really different. So there is a microphone here, so maybe you don't know, but mice can sing. They can sing in the ultrasound. I don't know if I can maybe give you an idea of what's going on. Okay, that's sorry. So that's the it's like a quick, but it's in the ultrasound. And so we are now looking at all these variations, and what Elodie found, sorry, that these mice they sing less, they sing differently with different call types. So now we are tracking these mice, and what is amazing at Pasteur is that these guys, they didn't know anything about mouse or anything about vocalization, but they were really great in terms of tracking system and images. And so we worked together, and it was really amazing. So at the canteen, we met at the canteen. And so we are also doing a lot of experiments because if you take if you discuss with people with autism face to face, it's okay. But if there are many people in the room, then they are, it's really a problem. So we want to change the mice to exactly see what is the problem for these mice and trying to find new treatments to improve this. So to find treatments, this is our strategy. Autism or autisms is very heterogeneous. Okay, and if we want to find a cure for autism or a treatment for autism, really need to stratify patients. So that's why we're doing a lot of genetics. We're doing a lot of search on biomarkers for autism. And then we can select some patients with synaptic mutations, doing new iPS cells, neurons, animal models, to try to better stratify the patient. That's why I am in the neuroscience department. Some of my colleagues, geneticists, they say, oh, autism is glutamate. And I think it's more complex than glutamate. So we are working on this, and to have the best molecules for these patients, that really like haute couture approach. So the perspectives, to have mutation is a rule. We have a lot of mutations. We still think about the genome like the American movie in the 50s where the good guys were in white and the bad guys were in black. The genome is not like this. It's not a pure genome when you have a mutation. It's full of mutations, and we can cope with that. So, but we have to work on this new way of thinking about the genome. So you will have different types of autism. Sometimes you have really nothing in the parents, and you have a very strong mutation that will lead to autism in the child. Sometimes you have parents will already have quite a lot of mutations or variations, common variants that will lead them to increased risk of autism, and their child will have autism. But this is a really complex picture, okay? And I think we have to be careful about trying to reduce the complexity of autism. So this is my way. I'm a big fan of autism, and I used to like that quote a lot, but since I work on autism, I really like it. I mean, I accept chaos. I'm not sure whether it accepts me. And I think we have to be careful in trying to reducing complexity. Okay, so we have to work together. That's, again, Pasteur is an amazing place to work together with the network of Pasteur. We're now a new project with other Pasteur institutes around the world. I'm the PI, the principal investigator of the genetics part of the largest European consortium on autism research. And the idea is to stratify patients and to see for each patient what is the problem. Okay, if he has sleep problems, if he has this kind of autism. So it's a lot of work. It's a lot of work that we have to do together. In New York, I work with the Simons Foundation is doing a great, amazing work on autism research. The ABIDE, the Autism Brain Imaging Data Exchange, there is a lot of data sharing in autism, and I think it's better and better. So why finding genes for autism spectrum disorder? My answer is to find the best environment. I showed you this picture with the phenotype called the genotype plus environment plus genotype environment. And I think in autism it's not like this, it's more like this. With some patients with mutation in neurics and neuroligin, some patients with other mutations. So if we have an idea of the G, what is the genetic part of this disease in patients, then we'll get the best environment. And one of the projects now at Pasteur is trying to understand how can we cope with these mutations. I found some parents, they have a very strong mutation. I thought, oh, but they should have autism, and they don't. So what's going on in their genome or in the environment? And I love that quote from Pasteur. The best doctor is nature. It cures 3/4 of the disease and never speaks bad of his colleagues. And I think we have to understand about what nature could have done for autism and about the genome and the environment also. Okay, one of my best environment, les amis, and again, I mean, I'm the guy who's speaking, but there are the guys who are working, and this work couldn't be done without Marion Le Boyer and Christopher Gilbert, who initiated this work a long time ago, before I was working on autism. And just to give you an example of the Pasteur Institute, this guy, Guillaume Dumas, he just arrived. He didn't know anything about genes, but he's a great guy in graph theory, and so I will teach him a little bit about genes, and he will give me a lot of knowledge about how the networks are working. Okay, that's the money. I have to thank a lot of people, especially the Fondation Bettencourt-Schuller, get the recipient of their share, and they really fund us a lot. And the Sfari Foundation is funding our working on the World Genome Project, and of course the Institut Pasteur. And I think that's a great place to be. It's not because you were there, Christian. I say that even if you were not. And I will thank the family, les familles, and I love this title. This is a social brain meeting organized each about 2 years. No mind left behind, and I think it's really interesting to have this point of view. And this is a cartoon of a father who has a daughter with autism, and on the T-shirt it's written, "I am unique, just like everyone else." That's my message as a geneticist. Thanks.